Okay, now for the flow of changes after the death of fish, several biochemical and microbial changes immediately begin to occur in the fish body. Since the fish is no longer alive, all physiological activities gradually stop, leading to quality deterioration and spoilage. Okay, for the next step is blood circulation stops. Blood circulation stops because the heart is no longer functioning. As a result, oxygen and nutrients can no longer be transported to the tissues and organs. Waste products also cannot be removed from the cells, causing the body condition to deteriorate. Next, stoppage of action of phagocytes. At the same time, the action of phagocytes also stops. Phagocytes are defense cells that normally protect the fish by destroying bacteria and harmful microorganisms. Once these defense cells stop functioning, bacteria can easily survive and multiply in the fish tissue.
Okay, oxygen supply fails. The oxygen supply fails because there is no blood circulation to carry oxygen throughout the body. The muscle tissues become deprived of oxygen, creating an anaerobic condition inside the fish body. Okay, next for oxidation reduction potential falls. As oxygen levels decrease, the oxidation reduction potential also falls. This means the tissue environment becomes less oxidative and more suitable for anaerobic biochemical reaction and microbial activity. Okay, next for respiration cases. Due to absence of oxygen, normal aerobic respiration cases, the fish cells can no longer produce energy efficiently through the normal metabolic pathway.
Okay, next we move to the glycolysis commences. In this process, glycogen stored in the muscle is broken down into lactic acid to produce small amounts of energy. However, this process is less efficient compared to aerobic respiration. Next, we move to the pH falls. As lactic acid accumulates in the muscle tissue, the pH falls and the fish flesh becomes more acidic. The decrease in pH affects the stability of muscle proteins and contributes to postmortem biochemical changes. Next, we move to the ATP and creatine phosphate level falls. At the same time, ATP and creatine phosphate levels continue to decrease because energy production has stopped. ATP is essential for muscle contraction and relaxation. Without sufficient ATP, the muscle cannot return to their relaxed state. Next, we move to the onset of rigor mortis. When ATP is fully depleted, the onset of rigor mortis occurs. During this stage, the fish muscles become stiff and rigid because the muscle fibers are locked in a contracted condition. Rigor mortis is one of the major post-mortem changes in fish.
After rigor mortis, protein denaturation begins. The acidic condition and biochemical reactions cause the structure of muscle protein to change and lose their original properties. This affects the texture, water holding capacity, and overall quality of the fish flesh. Furthermore, enzymes known as catepsins are liberated and activated. These enzymes are naturally present inside fish cells, particularly within lysosomes. Under low pH conditions, the enzymes become active and begin attacking muscle tissues. Next, protein breakdown. The activated enzymes then cause protein breakdown by degrading muscle proteins and connective tissues. As a result, the fish flesh gradually becomes softer, weaker, and less firm.
As a protein degradation continues, exudation and discoloration occurs. Exudation refers to the leakage and fluid from the fish tissue, while discoloration refers to change in the color of fish flesh. This change reduces the freshness and the appearance of the fish. This is bacterial growth. Bacterial growth increases rapidly because the fish defense system is no longer functioning and nutrients from the degraded tissue become easily available for microorganisms. The bacteria multiply quickly, especially if the fish is not stored at low temperature. Finally, Spoilage compounds such as methylamines, free lipids, and protein degradation compounds accumulate in fish tissue. These compounds are responsible for unpleasant odor, slimy texture, resinity, discoloration, and overall fish spoilage.